talk about our fungicide research trials and talk about some future work that we have that we will be doing this upcoming year. So a little bit of background on Boxwood blight. It was first described in the 90s in the United Kingdom, and then in New Zealand it was reported in 2002. And both of those were kind of described as two separate fungi, but we now know it's the same fungus that causes the disease. And then in 2011, we kind of found reports in North Carolina, Virginia, and Connecticut of the Boxwood blight, kind of all happening that fall of 2011. Today it's found in multiple states, but there's still kind of a large concern about the spread of Boxwood blight and moving it around. So that's a concern for the nursery industry. So why are we talking about Boxwood blight? It's a devastating disease, as you can see from this picture here, and maybe you have seen in the landscape. This one, you can see half the plant is defoliated. In really severe cases, the whole plant will be defoliated and people will have to rip that out of their landscapes or their plantings. But it's a popular plant because it is deer resistant. The deer don't eat it. So this is a high value for the landscape and nursery industry. And it's growing in popularity too for Christmas trees and decorations around the holidays. So what are the first symptoms we see before it gets to this stage? So the first thing we see are leaf spots, and I have kind of several pictures here of the leaf spots, and you'll first see them in the lower canopy first. So as you can kind of see these small black necrotic lesions on the leaves. And then here, some more examples of the lesions, and you'll kind of see the browning of the leaves, and they might be on the tips. There's usually this kind of black, dark black, brown margin too on the lesions that you'll see. Here's just some more pictures of lesions. You can see as the lesions age, the whole leaf becomes necrotic. And then the stem, you start seeing stem lesions as well, these black streaks on the stem. Another thing you can do when you're looking at the lesions is you can look for sporulation, which is fungal growth. So on this kind of white stuff you see on the back is the fungus starting to sporulate, producing lots of spores. So that's one key characteristic. Here's another picture. You can see kind of a close-up, once again, that white mycelium and sporulation occurring on the leaves. So when samples come into the lab, for me, that's the first thing that I look at, is to see if there's any sporulation, because it's an easy way for me to say, yes, this is Boxwood blight, without having to culture it. And then on the stems, you'll see the black lesions. So this is also really good to look for if the disease has progressed far enough. You'll want to look for the stem lesions because if the leaves have defoliated off, you should still be able to see stem lesions. Here's just another close-up, looking once again that dark black stem lesion. And here you can see that as it progresses, the leaves will brown, fall off, and you'll kind of have half of this dead plant. And then once it gets really severe, here's kind of a picture of, you can see just the whole shrub, a lot of the leaves have fallen off. So will your boxwood get blight? So there's different, all the Boxus genera are susceptible, as I have some of the ones that we're commonly hear about listed here. And the cultivars will all vary in their susceptibility and tolerance to boxwood blight, but there's not one specific species that's better than another. And so it's just important to make sure you're looking at what cultivars listed. So there's some different places to find that information. The nurseries will probably have some information on their tags and everything for the boxwoods. Also, Kelly Ivers, who is the predecessor to my boss, Sarah, she did some work looking at different cultivars and their susceptibility to boxwood blight, and that information is on the NC State Extension Ornamental Plant Pathology Extension website. if you want more specific listing of cultivars and their susceptibility and tolerance to boxwood blight. There are some additional box blight host plants. So Pachysandra, which is commonly also seen in the landscape, can also be a host for the boxwood blight. So the fungus can kind of live and survive and hang out in those plantings, even if you're not necessarily seeing the predominant symptoms, or maybe the plant's not affected greatly, but it can definitely still survive and hang out, and then of course spread to your boxwoods. And also sweet box is in the same family, plant family, so it can also be susceptible to boxwood blight. So just as people are planting or planning their landscapes, to be aware that these other plants can also be a host. So what is the pathogen behind boxwood blight? So it is caused by a fungus, the Calanectria. It thrives in wet, humid environments. It produces lots and lots of spores during one growing season, and it can also overwinter. It produces these structures called mycosclerotia, which is a hard mass of fungal mycelium, and those can survive for quite a while, and they will hang out in the soil. So just to go over the disease cycle, the life cycle. So once you have a healthy plant and once it becomes infected with boxwood blight, however the boxwood blight would be introduced, and then here you'll see the symptoms will develop, and then they will cause these lesions here. Lesions that will then begin sporulating. So this is kind of like a zoomed in version of pictures I showed earlier. You see that white stuff in the back of the leaves. So that's lots and lots of spores. So if you zoom in even closer, what you would see, and when I look in the microscope, I'm looking to see these rod-shaped spores here for the boxwood blight. And so this is many, many spores here, and this would be on one tiny lesion. So you can imagine if a whole plant has lots of lesions that are sporulating, that's quite a bit of inoculum that can be spread around. So then those leaves fall off, defoliate, they can hang out in the soil, they'll also produce that sclerotia, and so then the following season you would get the same disease occurring. So how does the boxwood blight spread? For short distances, it produces lots of those spores that can then be rain splashed. It has the overwintering structures in the plant debris, so those can then spread to the following season. Here's just some more pictures of spores. But for long distances, it really spreads by humans because the spores are sticky. They can be moved on shoes, equipment, clothing, and if the leaves also can easily get into clothing and everything, and those will have spores on them. So the infected material spreads the disease, and these spores can survive for a long period of time, for 5 to 7 years. So that's something to be mindful, especially if you are working with landscapers who might be going to one planting that has blight and then moving to another place that doesn't. Like you should really encourage them to be mindful that this you could easily be spreading this disease. So how do we manage this disease in the landscape? So the key thing is scouting, and then I'll go over some cultural and chemical control, and then also being aware of other problematic boxwood pests. That'll be helpful for when you're scouting, and then also controlling those diseases as well, so that way you can have healthy plants. So scouting is key, so looking for those lesions is very important before it's too late. So here's again some examples of some boxwoods that have been devastated by boxwood blight. So the main thing to do when you look for it is to look for spores on the lesions or any other visible pests when you're scouting. Look for the lesions, is there decline, make sure you know all those symptoms. The key things to look for are those characteristic lesions, and if it's sporulating, that really helps a lot, and looking for the black streaks on the stems. It's important to know the differences between all the symptoms for different pests, so that way you can identify it. appropriate disease. And then also if you understand what the disease is and what causes that disease, whether it's fungal, bacterial, or something abiotic, all of those things are going to be treated very differently. So it's just important to know the differences before you start applying chemicals. So the key thing for boxwood blight management is to avoid introduction. So buy clean plants by asking the nurseries about their practices, inspect the plants before planting. Some more tolerant varieties could still harbor the fungus, so it's important just to keep an eye on the plant after you planted it. Monitor for the disease. So if it had been sprayed with fungicide in a nursery setting or anything like that, that could delay the symptoms, but you still want to be scouting and keeping an eye on your plantings. It's very important to clean equipment and avoid using contaminated tools. And it's important when we talk about sanitizing tools that it's important that they are free of dirt and debris and they're clean, and then you sanitize. If you're working with any type of plant disease. So it's very important that, because if you're using a sanitizer on something dirty, it's not going to be as effective. And then also plant less susceptible cultivars, especially if people are doing new plantings. So here's kind of a list. There's a longer list online, but here's a list you'll see the more susceptible of cultivars here, the Justin Browers, and then there's the Green Beauty and the Nana that are more resistant or tolerant to boxwood blight. So there's kind of a big scale. So it's just important range as to what's susceptible and not to look into that before people are planting, and really encouraging people to plant less susceptible varieties. Here's kind of another list. As I said, you can go to the Ornamental Plant Pathology Cooperative Extension website, and you'll see a more detailed list of resistant cultivars. So other things for cultural management, to prune out diseased stems and rake up any diseased foliage that you see around the planting. It's important that you do not compost the diseased tissue because the spores could really still hang out. So it's important that you haul it away, unfortunately, to the landfill. But if you the compost, the spores could still be hanging out. So I don't recommend that. Also, if you're replanting, it's important to remove that kind of top layer of soil because the mycosclerotia and spores could be hanging out there. If you have an existing planting and you've raked all those leaves out and everything, you can mulch around the base of the plants to help bury that mycosclerotia and help kind of prevent some spread. As I said, cleaning tools is very important, and then wear plastic booties over field boots. It's important to minimize leaf wetness, so either through your pruning or choosing cultivars with a more open growth habit will really promote some airflow, and if it doesn't have that humid microclimate, the fungus isn't going to be as happy or as it's growing. So another thing is to promote airflow, you can maximize the space between plantings. Very important to avoid overhead irrigation and plant in areas that will dry out quickly. So usually in the photos that I was showing where half the plant is dead and the other half is still looking okay, some of that kind of depends on when the sun is hitting that plant. So half the plant is drying out quicker with after rain or dew. So then the disease isn't as bad on that part of the plant. So just something to be mindful as you're planting in the landscape. And then as far as chemical control, it's important to note that the fungicides will not get rid of the blight once it is present, but it can reduce some of the losses. And if you're using a preventative fungicide programs, will have to be sprayed throughout the season. So you'll have to do continuous sprays. And it's important to note that the sprays will need to be continued as long as the weather is warm in the fall. So especially like this last year, it was really, it remained warm for quite a bit into the fall. So that's just, you would have to keep managing the disease until it actually gets cold temperatures out. So a little bit background on fungicides. There's what are called multi-site fungicide or protectants, kind of like chlorothalonil. These have some pluses and minuses to them. So they can prohibit fungal spore germination. Their application tends to be by prediction or on a calendar-based schedule, and kind of when you think you need to spray. They kind of have, since they're multi-site, they can have a risk of harming non-targets or like phytotoxicity in some cases, because they're not acting on a specific part of the fungus. They're targeting a bunch of things. But they do have a low risk for resistance development. So because they're not acting on one certain pathway or whatever, resistance won't develop because they have hit several sites of the pathogen. Versus a single-site fungicide, what we call single-site fungicide, they're used to manage a number of phytopathogenic fungi. They minimize harmful effects to non-targets in the environment because they're focusing on one specific metabolical pathway of the fungus. So they're very specific. And they have a high level of protective and curative activity in some cases. So they're very effective. But their specificity fosters development of resistant populations with repetitive use. So with these, you can't spray. Usually the label only limits you the number of times you can spray per season. So it's important that you would use different modes of actions, different chemistries, and rotating the products so that way you don't get resistance in the fungus to that specific fungicide. So currently labeled single-site fungicides, there's Spectro, which is the chlorothalonil with the T-methyl. There's Torque, tebuconazole, which is a FRAC3 group. Medallion, which is a different FRAC group, different modes of action. Pageant, which is a mixed product, and Orchestra, also a mixed product. And both of those are better when used as protectants to kind of prevent. Here's another list of what's kind of used for chemical control. I guess some of these are for professional use, so these would be for your landscapers that have pesticide licenses that can go out and spray. And so here are some products to highlight, or as I previously mentioned on the other side, there's Torque, Medallion, Spectro, and Pageant, as I mentioned previously. There's kind of an updated list on the Virginia Cooperative Extension website has a lot of lists of different trials, and if you also go back to the NC State website, there is, Kelly Ivers had also done some fungicide trial work, so there's kind of some reports you can read for different products that work at controlling for Boxwood blight. So it is important to read the label to see what's, to make sure it's registered, and how to use it. And it's important to know that what FRAC group it is, and with those you're only allowed a certain number of sprays per season, and it's very important to rotate the chemistries, so to really know what group they're part of and to make sure you're rotating and limiting yourself to the maximum number of sprays you're allowed, and not going over that amount. So quickly, I'll go into a few other diseases and pests. So I wanted to touch on Volutella blight. So this one, the symptoms, the leaves turn light green and then they'll turn tan. The new growth might not be as vigorous. The infected branches will die back. So in some ways, it can maybe look similar. And you'll usually actually see this with boxwood blight. I think every sample we have that comes in has Volutella on it, which for me is kind of a pain because it's Volutella likes to grow a lot faster than boxwood blight in culture, so it's hard for me to isolate the boxwood blight. But this here, you'll see these orange or salmon colored spores, so you're looking, this is different than looking for that white that would be the boxwood blight. So kind of similar for Volutella blight management is proper pruning and thinning. Make sure you prune plants when the plants are dry. Remove all the dead branches and fallen leaves. You can try shaking out the plants to help remove leaves. And then the next pest I want to talk about is boxwood leaf miner. These form lesions or swelling on the leaves that may have a blistered appearance, kind of as you see here. And so I find that these, it can just look like a lesion, so sometimes it can be confused with boxwood blight, but then the leaves will turn yellow and can defoliate. So as you can see here, the lesions develop over time and they kind of age, so if you're not real familiar with what you're looking for, it might be confusing. So the leaf miner is the top insect pest of boxwoods. You see immature larvae of small orange fly. So when you break open a leaf, the easiest way to see is you'll see this orange larvae in the leaf here. And that's how you can tell if the lesion is caused by leaf miner. So typically the flies are seen in the spring. They lay the eggs and then the larvae will be feeding on the plant tissue. So they overwinter and will emerge as adults the following spring, and there's only one generation per year. So these are kind of hard to control because they are by the time you notice them flying around, they might have already laid their eggs, and then once the larvae hatch, there's not too much to do, but there might be a few, I'm not going to speak to it, but a few systemic insecticides that people could use if you're interested in that. But yeah, you can break open the leaves and you'll see the larvae squirming around in there. So another disease just to touch on is boxwood dieback. This is kind of a more of a new or newly reported disease in the southeast, kind of a lot of reports coming out of Louisiana. It's noted primarily for death or dieback of part of the plant. And so it kind of seems right now that the management options are limited until they kind of know more about this disease and the pathogen. It is caused by Toxtricum or a fungus, but you can go ahead and kind of manage it similar practices to boxwood blight. And then Phytophthora root rot, it's found in the soil. It's a fungus-like. The symptoms are the leaves turn yellow as the plant declines. The roots so if you see a declining plant, you can also kind of dig up the roots and you'll see that the roots are dead, they're dark and rotten. The bark will peel and kind of rots at the crown. So the main thing about Phytophthora root rot is it loves water, so plant in well-drained soils to kind of minimize maybe damage due to Phytophthora or its survival, but and avoid overwatering your plant. Make sure you buy disease-free plants. Once Phytophthora is found in the soil, it's pretty hard to get rid of it. So you would probably not want to replant in that same spot again if your plants are severe and you have to rip them out. Also, you really need to look into what products you can use to treat Phytophthora because it's not a fungus, but it's fungus-like. So the management practices are going to be a little bit different for chemical control. And now I will go into some research updates that we have. out of our lab. So the first, I'm going to talk about a couple different fungicide trials, some looking with traditional products or conventional fungicides, and then I'll talk about host plant defense inducers. You might have known them as SAR inducers, systemic acquired resistance, and I'll give more explanation on those. So the host plant defense inducers is kind of this idea, and they're all in the same frat group of mode of action of group P, and then they're kind of all with this new name lumped into the host plant defense induction group. So the and some of these products here are Actigard, Lifeguard, Regalia. You might have heard of those products. Sometimes they're more popular, maybe in organic settings. But the idea behind these products is that they might kind of give the plant a boost or activate some of its defense genes. So the idea is you could maybe apply this product and then the plant its defenses would be woken up, and then it might be better able to withstand the disease is kind of the idea behind it. And so this first product, the salsalate-related product here, that would be like your Actigard. This plant extract would be like your Regalia from giant knotweed. And then this microbial, or looking at a bacillus, is your Lifeguard. And so I'll go into, I have some data to show from kind of two different trials. The first ones I'm going to show are just looking at different comparison of plant host defense inducers. And we were looking at different application timings, one with just an application before we inoculated, and then we also did once before and then followed up with 2 other applications to see if the number of applications had an effect. And we also did 2 different types of applications. We did a drench and a foliar application. And then the second trials I'm going to talk about include looking at some of your conventional fungicides, looking at SDHIs, which are frat group 7, and your DMIs, and that's frat group 3. When we do our trial, we apply with a backpack sprayer, and we apply do our trials with both Justin Browers and Green Velvet. So Justin Browers is highly susceptible to Boxwood blight, and then the Green Velvet is supposed to be more tolerant or have some resistance to Boxwood blight. So we would kind of like to see the comparison of what's happening in each system. And then some of our trials we did inoculate, so we sprayed the fungal spores right on the plant to really promote disease. And then we also did a non-inoculated trial where we were at a grower site, and we had plants in the field that we treated. And those all already had Boxwood blight, but kind of at your natural levels of inoculum, nothing. We kind of put a lot of the fungus out there and get lots of disease when we do trials here in Mills River. So our potted trials that we did, so we inoculate with spores. So what we do is we'll spray the spores on the plants here. We kind of group them together when we're inoculating. Normally we have them all spaced out for when we're doing our spray trial. But for the 24 hours after we inoculated, we group them together and we cover with plastic to really promote that humidity and get the disease to take off. And then we have them under shade cloth and we water with overhead irrigation. So we got lots of disease, had no problem with the amount of disease. So the first thing I'll talk about is this year, just looking at the plant host defense inducers on the Justin Browers. So this is this is the highly susceptible cultivar. And the main thing, we had different treatments here. So we were looking at Prophyte, Actigard and Lifeguard, which are kind of those plant host defense inducers. And we were looking at either one time where we sprayed before we inoculated, or 3 times, which was the once before we inoculated, followed by 2 other sprays. And we did foliar and a drench. And then we always do our untreated control that was inoculated. And then we also compared to Kenja, this conventional fungicide here. And so the ones with gold stars were the only ones that had significant differences from the untreated control. So they didn't necessarily have differences to the other treatments, weren't necessarily significantly different, but they were from the untreated control. So some things that were interesting to us is that the Actigard foliar spray, followed by Actigard at a drench treatment, and at the drench was surprising. So we are interested to look into Actigard more to see if it still had quite a bit of disease, as you can see here, percentage-wise, but it still, because there were differences, it's something we want to investigate further. But as you can tell from this graph, here still conventional fungicide, Kenja here, provides much more control of the disease than some of these other products. And then if we look at the same trial on the green velvet, which is less susceptible variety, the thing to note here is overall we have less disease than we had on the we don't see as high of incidence of disease. So on the previous slide, the untreated control had about 90% disease, and we have a little bit lower here. Here we didn't see really any differences between the products, but the only one was that the Kenja foliar had significantly less disease than our untreated control. And then we also did a trial on potted plants again, kind of looking at more conventional with that Actigard. So here we have, once again, our untreated control. And this is the mean incidence of blight. So what we do is we take so many ratings. We take a stem, we count how many leaves out of so many have blight, and we take multiple counts per plant to see the disease level. So here we have on our untreated control, we had almost 90% incidence, and then you can see that Kenja at a drench and then Actigard at a drench had less disease than the untreated control, but still had quite a bit. And then Kenja as a foliar application, along with Pageant and the propiconazole, and these are all conventional fungicides. So the Kenja is frat group 7, the propiconazole is 3, which is a DMI. The Kenja is an SDHI, and then this Pageant is an SDHI with a QOI mix. Yeah, but as you can see here, just the SDHI had less, not significantly different, but had less disease than the Pageant. So it kind of makes you think that maybe the SDHI is doing a lot of the work. And then here we look at the inoculated experiment on green velvet potted plants. So this is the other cultivar that is less susceptible. And here we didn't see, so here we had some of the treatments, the Kenja and the Actigard didn't have, were not significantly different from the untreated control, and overall we had less disease because, once again, it's the less susceptible cultivar. And then kind of the same trends, this Kenja, the SDHI, propiconazole, a DMI fungicide, and then the Pageant here, which is an SDHI and QOI mix, provided control against the Boxwood blight. So kind of these products, you'd have to check. don't think they're registered for Boxwood blight because we're just testing to see kind of what chemistries work to control for the disease, but it kind of shows that the conventional fungicides kind of help manage the disease, and that the SDHI and DMIs work at controlling the disease. And then we also did this same trial. We did it at grower site. So these are on the potted plants, and we didn't inoculate this. It just had natural inoculum. And so here we saw that kind of similar trends, that we still had a fair amount of disease. So the Kenja as a drench and the Actigard as a drench still had less disease than the untreated control. And then Kenja and propaganazole as a foliar applications help manage the disease. So what else are we continuing to do for future research? So we are collecting a bunch of isolates. So we would like it if you see Boxwood blight or find a lot of it to send us just your plant samples, because that way we can isolate the fungus from those samples, and we can use it for some of our future work. So Sarah has a graduate student who's screening for SDHI fungicide sensitivity, since a lot of SDHI fungicides that FRAC group 7 are used to control for Boxwood blight. We want to start monitoring the fungus for any sort of resistance. And first, we just need a lot of isolates to just determine, to look at that work to make sure that it's still sensitive to the SDHI fungicide. And that's stuff we do in the lab. In vitro, we put some of that fungicide in media, and we watch, see how well the fungus grows or doesn't grow, hopefully, on that media. And then we will also continue to do fungicide efficacy field trials, specifically with SDHI and some other conventional products, just to continue to get a sense for what works to control for Boxwood blight, to manage it. And then we'll continue to work into field trials for the plant host defense inducers. So to kind of investigate those products more, to see maybe the timings or the applications, to how we apply them versus drench versus foliar. Since we did see some differences with Actigard, we'd kind of like to look into that more as well. And we're also collected a bunch of plant tissue at the times that we applied these products. So what we can do with that is in the lab look at gene expression to see if certain defense genes were actually turned on by these products, or if these products are having any effect on the plant. So that's some future work that we get to do here in the lab. So just real quick, some resources. There is the Plant Disease and Insect Clinic on campus to help identify any diseases or insects you have issues with. The NC Cooperative Extension website for ornamental plant pathology has a lot of the different handouts and stuff I talked about. And then the Virginia Tech website and the Connecticut Agriculture Experiment Station also have a lot of information. And they were just kind of also on the forefront of Boxwood blight. So there's a lot of information on their websites. And the Virginia Tech website has specific guides for landscaper, nursery workers, or anything like that. And it's different tailored publications if you are looking for different resources to give to people. And with that, I will say thank you. Here's a picture of the lab, the crew we had this last summer who did a lot of the work of raiding our boxwoods out in the fields. Alejandro on the end is a student from Peru. He will be doing he does a lot of the boxwood work, so he will be back in Mills River this summer, continuing these projects. And then if you have not met Sarah Villani, that's my boss, and I work for her, so she is the expert on, or becoming the expert on Boxwood blight. So with that, I will take any questions.
So we had a couple of questions down here in chat. The first one was, how deep of a mulch layer is enough to cover?
I would say you would probably want to add enough mulch without it being too much to let harm the plant. But I mean, I would think the thicker the better, because the idea is that you're going to bury those microsclerotia or spores from shooting out. So I would say the thicker the better, but obviously you don't want anything too thick because you don't want to affect your plant in any way.
And then the next question, folks were wondering if you would send out your PowerPoint, but we're recording this and we have a place where all of our webinars are posted. So this will be posted there and I'll send it out to everybody.
Okay.
And can you unshare your presentation real quick, and then I'm going to share a couple of things with folks.
Okay. Yeah.
So this is the Virginia Tech website that Rachel was talking about. If you scroll down, you'll see that they have the different situations, whether you're dealing with a commercial nursery or a residential boxwood grower. And I found these really helpful in working with growers. They have a lot of great information, but they'll be great for homeowners or anybody else who needs that. I think probably the most exciting thing that I learned last week at Green and Growin' in Greensboro, put on by the North Carolina Nursery and Landscape Association, is that Saunders Brothers has a new tolerant boxwood variety. It's supposed to be tolerant for boxwood blight and resistant for boxwood leaf miner. So that's pretty exciting. It's not going to be available till 2020 though. So we still have a little time before it's on the market. And as we learn more information, Danny, Amanda, and I will be sharing this with you guys. They haven't really given us anything other than they've developed it. It'll be out in 2020. We don't really know any specifics. Obviously, they're nervous to keep it proprietary, and they're not sharing a whole lot of information at this time. And then the only other boxwood thing that I wanted to share is a new pest for boxwoods. This one's not in the United States yet, but it is a moth that, of course, the caterpillars will feed on the boxwood. This is called the box tree moth. It's in all over Europe. It came into Italy from China and then sort of spread throughout Europe in about 20 years. And recently it was found in, I believe, Ontario, Canada, but it hasn't been found in the United States yet. And they haven't found a population of it in Ontario. They've just found, one or two moths here or there. But it always is a good idea to keep an eye out for things. So that's the adult, and then here is the larvae, obviously with the webbing, and it'll cause frass and chewing. So we can keep an eye out, and this is really going to be the only pest that's going to cause chewing damage on a boxwood. So keep an eye out for it, and hopefully, fingers crossed, we won't see it. I did see that we had one other question, so let me go back to that. Stop sharing, go back to chat. So then the question was, is boxwood blight widespread over the North Carolina counties, or is it concentrated in just a few counties?
I don't know. I was trying to find an updated source list, but I think that it probably is more isolated to areas that would just have more boxwoods in the landscape. However, it can be found anywhere, I think, at this point. So it's within North Carolina. But obviously you want to avoid movement. And I think if you're like going to different states nearby, you would maybe need to check to see if they have boxwood blight or what they're doing to manage it too. If you're looking at that, look at their Department of Ag websites to make sure you can move material. But yeah, I think within North Carolina, it's probably widespread.
And Paige mentioned that they have it in Watauga County and it's been confirmed by the lab. And I know that I've seen it in different locations around the state. So I'm pretty sure it's pretty widespread around the state by now. Any other questions before we end for the day? Just give it a moment on the chat box.
The question was, what's the current status of the Department of Ag as far as monitoring? I don't think that it's not any sort of like restricted pests, because I think it is pretty much found or common at this point. So I don't know that there's any ongoing. I don't know the answer to that if there's any ongoing, but when I was just trying to look up kind of the status to see what states it was all, where all it's been found. I just noticed that other states might be where it's not as prevalent, might be monitoring, but I don't think, I don't know that anything's going on right now for North Carolina.
In the nursery industry, they're monitoring it heavily. The nursery inspectors come around twice a year, monitoring anybody who has boxwoods, and you have to have a specific certificate in order to be able to ship out of state. As far as landscapes go, I don't know how heavily they're monitoring the landscapes. Danny, you know about that?
I'm not aware that they're monitoring it in landscapes.
Thank you. And so Paige asked another question, are they monitoring individual shipments or are they just monitoring the nursery itself? So they'll go around twice a year monitoring nurseries, but if a grower is interested in having a specific shipment monitored, they can call their NCDA nursery inspector and those folks can come out and monitor the shipments. I actually talked with a grower last week at Green and Growing who is getting ready to ship out a shipment and doesn't want it to leave until it's specifically checked. So I talked with the NCDA person this morning and they're gonna go out there pretty soon to get that checked for him.
Yeah, that's what I'd say.
Great. Thanks, Jean. We're happy that you found it useful. Alrighty, well, if there's not any other questions, we'll go ahead and end for the day. If you think of any questions after you get off the webinar here, you can feel free to email me and I can send over your questions to Rochelle. And I will be sending out the information on how to find this and all of our quarterly webinars. So thanks again to Rachel, and thanks again everybody who joined us for the webinar. I hope you guys have a great week. Thanks again.
Thank you so much for having me.